So you start with hematopoietic stem cells, which can be identified by Lin negative, SCAR positive, and KIT positive, right? And these cells then further differentiate into one lineage or the other. For example, if these cells have to give rise to T cells, they express these markers, CD3, right? Along with either CD8 or CD4. The cells which express CD3 and CD8 are cytotoxic T cells. The cells which express CD3 and CD4 are called helper cells, right? And they develop in thymus. Okay, so similarly, B cells, they express on them this marker. Rather, these one of these is B220, another is CD19. Okay, once these cells express these markers, you start calling them as B cells. And then you also have these cells which express based on high or low level of these two molecules, MAC1 and GR1. So you call them as either macrophages or granulocytes, right? Macrophages express more of MAC1 and less of GR1, and reverse for the granulocytes. Megakaryocytes express this CD41. They eventually give rise to platelets, and erythrocytes are identified based on the expression of this molecule that we call as TAR119. Okay, so now this slide actually basically tell you something about the activation status of lymphocytes when they are in their activated stage. Okay, so you have these lymphocytes which in which basically the nucleus covers almost all of it. Okay, so there is very small volume of these cells which is not having this nucleus. Okay, these are small cells, and in inactive stage, so they basically have a very big nucleus. Once these cells started getting this process of activation, so once they are activated, so then almost all of the cell cytoplasm is occupied by these molecules, rather these subcellular organelles that we call as endoplasmic reticulum. And the reason is because these cells produce a lot of cytokines. Their protein biosynthetic machinery is too rapid. Okay, and so almost all of the cell is occupied by ER. ER, has a boundary which is actually very diffused with the nucleus. Now, actually, this slide talks about how you can identify these different cell types based on flow cytometry. Okay, and just by looking at the FACS plots, which you obtain using some cells from that particular lymphoid tissue, so you can make an assessment that this organ, this lymphoid organ, might be what. Okay. For example, so if you get a FACS plot, which is something like as this. Okay, so you are basically getting on live cells, and each one of those dots that you see here represent one cell. Okay, so for example, these are this is one cell, this is one cell. There are so many of these cells here, so many of these cells here. And when you have actually these areas which are actually red and greenish and bluish, so that actually tells you the density. That means there are a lot of cells in this area, less of those in this area, and very less in the periphery. Okay. Now, why is it important? Because see, earlier these cells were different, rather identified based on their morphological characteristics of nucleus and the cytoplasm. So you use some dye, and how basically nucleus is stained and how cytoplasm is stained. So you can label them as neutrophils, monocytes, lymphocytes, eosinophils, and so on and so forth. Okay. But using flow cytometry, you can actually define a given subset of these cells in that particular population. Okay. Now, how do you interpret these FACS plots? So, for example, it's written CD4 and CD8. CD4 is written on y-axis and CD8 is written on x-axis in the first FACS plot, right? So this is not as such this molecule CD8 or CD4. You are using an antibody which recognize these molecules. Okay, so essentially on this axis you are looking at anti-CD4 antibody, which is fluorescently labeled, and on this axis, so you have an anti-CD8 antibody, which is fluorescently labeled, but the fluorescence for this antibody is different, and for this antibody is different, because if you have the same fluorescence, so you cannot really distinguish these populations, right? Now, depending on these lines that you draw in between, these FACS plots, these are called FACS plots. So you can label them as positive population and negative population. Same is the case with, for example, this line here. So something which is positive for this marker will be above this axis. All these are positive for CD4. And below this axis, so you will call them as negative for this marker. Okay, and if you use this line as the defining boundary, so these are positive, these are negative, these are positive, these are negative for this particular marker. Now, depending on what do these cells express, okay, so you have almost 10-11% of these cells which do not express any of the markers. Express CD8. So these cells are called double negative, and because they are coming actually from lymphocyte population. So these are lymphocytes, okay, but they do not express any of the markers. They are CD8 or CD4 negative. Now, in this quadrant, so these different sections are called quadrants: Q1, Q2, Q3, Q4, something like that, right? Now, in this region, so these cells express CD8 because they are beyond this axis, this vertical line here, right? And the cells on this basically below this axis or behind this axis, they do not express anything, or at least this marker. So you have CD8 positive, but CD4 negative. Okay. Similarly, in this in this gate or in this quadrant, so you have CD4 positive, CD8 negative. Okay, which is close to 5%. And in this area, in this quadrant, so you have almost 70% of these cells which are double positive. So they express both CD8 and CD4. Right. Based on the staining characteristics, so you can actually define these populations as positive for one marker or negative for one marker. Okay, so for example, in one of these things, I said lin negative. Lin negative means, for example, if you use an anti, okay, so these cells will not come in any of those gates. Okay, so they will be not expressing lin, but they will be positive for SCA1 and they will be positive for KIT1. Now, what are these markers? These actually markers are expressed on differentiating hematopoietic cells. Okay, and remember, so when cells divide from one lineage to the other lineage, one a precursor population to one phenotype or the other, right? So this is a very dynamic process. So they lose expression of some of the markers and they upregulate some of these markers. And depending on that, you define them either to be of this particular cell type and or the other cell type. Okay, so now let's get back to the same. Okay, now we are still on this first panel here, where 4 and what are CD8 and what are CD4? What are how do you broadly classify these cells as? Are these B cells? No, these are T cells. Okay, some cells are CD8 positive and some cells are CD4 positive, and some cells are double positive. Right. Now, if you look at the expression of CD4 and CD8, and you find that there is a significant proportion of your lymphocytes which express both of these markers. So invariably, so this sample was prepared from thymus. And why? Because thymus is the site where development of these different T cell subsets, CD4 and CD8, takes place. Okay. Essentially, basically, how these cells start their differentiation, and not exactly differentiation, but development. You call them as development, because this is predominantly an antigen-independent development of these cells. So you start with double negative cells. These cells then become double positive. Now, depending on whether these cells are coming in contact with MSC class 1, so they will become single CD8 positive. If they come in contact with MSC class 2, so they become CD4 positive. Okay, so these are again broad steps in the development of your T cells. Okay, and as soon as you see something like as this, where you have a lot of cells which express both CD8 and CD4 on their surface, so you can say that these cells must have come from thymus. Thymus is a primary lymphoid organ which is present in your thoracic cavity on top of heart, right? And the site for development of your T cells. Okay, now from the same organ, so if you actually stain for this marker, which we call them as CD3. CD3 is a pan T cell marker, so it should be expressed on almost all of the T cells. All right, and you can see that almost all of these cells on y-axis actually express this molecule. Okay, so you can say that these express low level. They are not really negative, but nonetheless, these cells do express some level of CD3. and almost 75% of these cells actually are expressing quite significant level of CD3. Now, this is a T cell marker. And if you use another marker that we call a CD19 or B220, okay, you don't find any cells in thymus which are CD19 positive, because thymus is the site where you do not find any B cells. B cells are present in bone marrow. They complete their process of development in bone marrow, and once they have passed through these processes of development, so they get into periphery. In lymphoid organs, okay, in secondary lymphoid organs rather, lymph node and spleen, so you find a lot of B cells and a lot of T cells, okay, and so that can be identified if you talk about B cells as based on expression of this molecule CD19. So now if CD19 is absent in a particular organ, so you would again want to call this organ as probably is thymus, because you don't have any B cells here, right? So now, based on these different cell types that you recover from a particular lymphoid organ, so you can actually try to say whether this is thymus, this is bone marrow, this is a lymph node, and so on and so forth. Okay. Now, in this panel here, so I want to tell you something more, which is basically, again, you prepare some single cell suspension from these lymphoid organs, right? On one axis, you are using an anti-CD3 antibody. On another axis, you are looking at anti-CD4 antibody. The expression of CD4 molecule, which will be detected using this antibody against CD4. And T cell marker using this antibody against CD3. Again, you get these cells which do not express either CD3 or CD4. In this case, so you have these T cells positive, right? So the lineage marker for CD3 is positive on this axis. And here, so you have double positive cells. So these express CD3 as well as CD4. And here, so you do not have many cells which express CD4. Now, you already know that CD4 cells actually came from a subset of CD3 positive cells, right? Yes or no? CD3 is a pan T cell marker, and CD4 is present only on a subset of these cells. Now tell me, so what are these cells then in this Q3 quadrant? So you have Q1, Q2, Q3, Q4. Okay, so in Q3 quadrant, what are these cells? Just by looking at these two markers, can you make out anything?
So cytotoxicity cells.
It's positive. Okay. So, and something like this, this staining actually would be present in your secondary lymphoid organs. Okay. So this slide basically makes an emphasis that based on the staining characteristics of your cells of the immune system, so you can find out, or you can actually make an assessment whether this is thymus, whether this is lymph node, whether this is bone marrow, whether what this organ might be. You just have to choose the right panel of markers, right? And these plots are bi-exponential FACS plots. Okay, so you are looking at two markers, one, and you divide them into these quadrants. In this area, so you get double negative. Here you get double positive and single positive for whatever the marker you are looking at. And this organ where actually you find more of these double positive cells for CD4 and CD8. You don't find something like this in any of the secondary lymphoid organs. Okay, you also do not find this in bone marrow. So this has to be the thymus. Thymus is the site where T cells development takes. Any question on this? There is some comment in chat box. Can someone read it?
Sir,
Yeah.
so Mridula has written that as we see in the plot at top right has cut off around 10 to the power 1 for both CD19 and CD3. However, in the bottom plot, the cutoff is between 10 to the power 1 and 10 to the power 2 for CD3 and around 10 to the power 2 for CD4. So how is this cutoff lines distinguishing positive and negative estimated for each fax plot?
So that actually depend on whether you have a very brightly staining antibody or not so antibody, which indirectly will be reflected based on the fluorescence you are using. There are some fluorescence which are very bright and some fluorescence which are not that bright, they're dim. Okay, so how do you basically decide? That is based on these staining controls. For example, you have one sample where you do not add any of the antibodies and then see a sample, so you should not have any positive cells. And then you try to set a gate, right? And also it depends on what concentration of antibody you have used. And that these are the things that you need to optimize before you perform any staining, any flow cytometric staining in your analysis. Okay, so you have to use different concentration. For example, 1:50, 1:100, 1:500, and then find out at what particular concentration of your antibody, so your positive population is positive and negative population is negative. Right. Yeah.
Sir,
Yeah.
so I have a doubt in the third plot. So in the T cell development, at what point of time the T cells are showing CD3 positive cells?
CD3 will be present on these cells right from the word go. Okay, when they are actually when they have already left bone marrow and they are now present in thymus. So CD3 is the first molecule which comes up on their surface.
So, sir, what does the CD3 and CD4 negative cells showcase in this plot in the first quadrant?
Well, because this is coming from secondary lymphoid organs, so obviously these have to be B cells. Or maybe some of the innate immune cells. Right?
Fine, sir.
Okay, so we referred to in one of those early slides was these monocytes, when they get into a particular tissue site, okay, so you start calling them differently. Okay, so in blood, so these cells are called monocytes. Once they get into tissues, so you call them as macrophage, right? Now, depending on what area, what part of your body, so you find these cells in, so they are named differently. Okay, and they can actually, if you talk about macrophages, so they are labeled with this marker CD11B, MAC1. That's the other name of this molecule. Okay. Now in gut, so these are called intestinal macrophages. In lungs, these are called alveolar macrophages. If these cells are present in connective tissues, so you call them as histiocytes. If these cells are present in liver, you call them as Kupffer cells. If you find them in kidney, mesangial cells. If you find them in brain, you call them as microglial cells. And obviously in bone marrow, you call them as osteoclasts. Right? So depending on what location these macrophages or you can say monocytes are present, so you call them as with different names. You just have to remember these things. That if you find these macrophages in brain, so you call them as microglial cells. Kidney, mesangial cells. Liver, Kupffer cells. Histiocytes in connective tissues. Right? And so on and so forth.
Excuse me, sir.
Yeah.
Sir, could you please go back to the previous slide? Yes, sir. So, sir, I just wanted to ask that in all these organs, do macrophages have the phagocytic activity?
Yes.
In all of them?
They have. Well, that is very canonical for macrophages. Okay, it's just that they start expressing some additional markers. And they actually provide surveillance in that particular area.
Right, sir.
Yeah. Okay, so other relevant cell types, one of them actually is dendritic cells. And we also talked about that the person who discovered these dendritic cells was acknowledged for his contribution with the Nobel. Okay, now these dendritic cells are the cell types which connect particularly the innate and adaptive immune responses by engaging the cells of your adaptive immune system, particularly T cells, and very critical for initiation of T cell responses. What do they express on them, on their surface? This marker that we call as CD11C, in addition to CD11B. Right? Now, that is why actually some people still call them as that these are dendritic cells are nothing but modified macrophages, because they express this CD11B marker. In addition, they started to express CD11C. Okay. Now, dendritic cells come in different flavors. Okay, so you call them as myeloid DCs based on these two markers, plasmacytoid DCs, which produce a lot of type 1 interferons and are very critical initial responder following a virus infection, plasmacytoid dendritic cells. Okay. So you also find follicular dendritic cells in your secondary lymphoid organs. Although they do not express a lot of MSC class 2 on the surface, nonetheless, they can present these antigens and can activate your T cells. Now, you would want to say that if they don't express sufficient MSC class 2, so how can these cells be called as antigen presenting cells? The mechanism for their antigen processing and presentation is different. Okay, this occurs through take of these immune complexes. Okay, so antibody is bound to antigen, and this antibody, you know, the structure of antibody is like this Y-shaped structure. On this side, you have the antigen bound. In the stalk portion, so you have this Fc portion, right? Fc receptors actually are predominantly present on these follicular dendritic cells. And because of this process, so they can take up these antigen, they can process and present to T cells. Okay, these are called follicular dendritic cells. Now, there is another cell type that you see predominantly at mucosal sites. Okay, and these are called M cells. As M stands for membranous. It's just the architecture of these cells. Okay, these are actually modified epithelial cells in your gut or at the mucosal sites, and they appear like membrane. Okay, so this is how these cells look like. So you have this intestinal wall, small intestine, right? And M cells are like this. They are membranous cell types. And as you can say that these are all those epithelial cells. And in between, so you have some modification in these epithelial cells, and they facilitate the entry of these antigenic components coming from different microbes or even the food that you are eating. Okay, M cells. Right beneath these M cells, so you have a lot of your immune cells like macrophages. Okay, and then you basically make these pious patches where you have T cells, B cells, and so on and so forth. All those things are present. Okay, so just by looking at the structure of your intestine, you can find out that this is the portion which actually is more. You can actually anatomically identify M cells from the rest of the epithelial cells. Okay. Now, epithelial cells as such are very bad in inducing immune response. They don't generate any immune response. But in your gut, at certain places, so you have these M cells, and these M cells can sense the presence of these antigens, and they can engage your immune responses by activating B cells and T cells. And then they produce antibodies and their effector molecules when you talk about T cells, right? So this is also to emphasize that there is one particular cell type which is present in mucosal sites and is crucially involved in induction of immune responses in your gut and other mucosal sites. All right, so okay, so now this slide actually summarizes. Now there are quite a few things from the innate immune responses that we have to talk about, and one of these things actually is the complement system. And as I said in the beginning, one of the major pathways for activating complement system involves the antibodies, the response of antibodies. Okay, so if you don't have antibodies, so you don't activate that very important pathway of complement system. So that is why once we are done with the antibody part, so we'll talk about complement system. Okay, this is not to say that complement system actually is part of your adaptive immune responses. No, so this is part of your innate immune responses. But antibodies are involved in triggering the activation of complement system. Okay, so that is why we are going to cover it later. Once we have discussed the structure of antibody molecule, because based on the structure, you would know why these antibodies can also activate complement. Okay, before that, so basically this slide talks about the sequence of events. For example, following a virus infection. Okay, so on x-axis you are looking at days after viral infection. So what happens at different days after you gotten infected with a virus. Okay, and on x-axis, or rather on y-axis, you are looking at the magnitude of these different molecules or the number of those cells you can find out. Okay. Now, initially after infection, so the first thing, particularly when you are basically talking about the viral infection, the one thing which is very critical and is induced first is these interferons, interferon alpha and beta. These are called type 1 interferons. Okay, very quickly following this infection, so type 1 interferons are induced. After that, you have the activity of your NK cells. So they start expanding in numbers, and they reach a peak, and eventually there is contraction, right? And then you're left with a pool of these cells. Okay, and this shaded area actually is the virus that you are able to detect from that organ. Okay, for example, if you get respiratory infection, so let's say this shaded area represent over a period of time how much of this virus will be present in your lung tissues. Okay, or if you have skin, so basically you're talking about in that local area, so how this virus is growing. Okay, so starting from day 3-4, so you basically now you have initially activated your innate immune cells. These innate immune cells, they have taken up these viral antigens at the infection site. So now they have migrated from that infected site to the draining lymph node, and we already have talked about the concept of draining and non-draining, right? So in draining lymph node, they engage with specific receptor expressing T cells, right? And once this engagement of your APCs presenting these peptides through MSC class 1 with the TCR, a specific TCR expressing T cell takes place, and then you have two more sets of molecules that we'll talk about at some time point. So you activate these cells, following activation, they proliferate, they divide, and they expand in number. Okay, as is shown by this upward curve with black line, right? And now with the expansion of these antigen-specific or virus-specific cytotoxic T cells, so you also try to control the growth of virus, right? And later on with this red line, whatever is shown is the level of your antibody response. And these antibodies obviously are produced by your activated B cells. Okay, so now you know the sequence. Okay, the sequence is basically first is type 1 interferons, then NK cells, then your virus-specific cytotoxic T cells, and then your antibody response. And a concerted activity of these cells tend to control this virus buildup, right? Okay, so type 1 interferon. So this is actually a brief summary how type 1 interferons act. Okay, you have multiple units of these receptors, type 1 interferon receptor. Okay, so once ligand is bound, once you basically and what produces type 1 interferon? So all kind of cells which are infected with virus can produce type 1 interferon. Receptors for this type 1 interferons are present again on all kind of cells. They are ubiquitously present, right? Let's say the cell is infected with this virus, it produces interferon. Now, these interferons will act on the same cell, and this is called autocrine, a mechanism of action. They also act on the cell which is not infected. Okay, this is called paracrine function. How do they act? By the dimerization of these receptors in the membrane. And because of dimerization, now this type 1 interferon can bind to these receptors more efficiently. This interaction of type 1 interferon with the receptor units induces phosphorylation of the amino acids which are present in the cytosolic tail of the receptor. Okay, K is nothing but basically suggesting addition of phosphate. Okay. Rather, these are these kinases basically which get recruited, and these kinases add these phosphate group onto specific amino acids, tyrosine, for example, serine, threonine. Okay. So those amino acids are phosphorylated, and once these amino acids are phosphorylated, so now you create docking sites for other molecules. Okay, so for example, these stats which are normally present in cytosol, so they will come and they will get recruited here, right? You generate these stats. Okay, once these stats are deposited, or they are basically scaffolded onto the cytoplasmic tail of this receptor, so they are again phosphorylated in their tyrosine and serine residues. As a result of it, now it will dislodge. Okay, it will form stat dimers. Okay, so you make these dimers. These stat dimers, they basically migrate to or translocate to nucleus, right? In nucleus, they bind to the promoter region of these different genes, and they bring about the transcription of these genes. And many of these genes, which are induced as a result of type 1 interferon signaling, we call them as antiviral genes. There are many of those. Okay, for example, ribonuclease L is one, oligoadenylate synthetase is another one, right? IFIT proteins are synthesized in these cells. And once you have all these proteins synthesized, and where would this synthesis take place? In ribosomes, right? So now these molecules, these proteins, they have this antiviral function. Okay, for example, if you have ribonuclease L activated, so this is going to degrade mRNA in the cell, and as a result of it, so the virus will not be able to divide in these cells. Degradation of viral genetic material, right? And when protein synthesis is inhibited in these cells, so you achieve what we call as an antiviral state. This is how type 1 interferons work, to block antiviral activity. This can occur in an autocrine manner, but more efficiently in a paracrine manner. Is that clear how type 1 interferons work? Yes? Okay, I'll give you a couple of minutes, or maybe less than a minute. You can read it. Just go, just read this slide, whatever is written.
Sir, could you please revisit the previous slide?
First read it, and then we'll get back to this same slide. Done? Let me know when you're done.
Done, sir.
Okay, so yes.
Sir, can you please explain the following pathway after translation till antiviral state once again?
Well, you synthesize the specific molecules, for example, oligoadenylate synthetase. Okay, so now as the name suggests, so this depletes ATPs, okay, adenyl, right? So the synthesis of all these molecules, the energy currency is depleted in these cells, and when these ATP molecules are not available, so the protein synthesis within these cells will be hampered. Now, because there is no protein synthesis in these cells, so whatever the virus has entered into this cell will not be able to make more copies. That's one. Whatever the virus has already entered, its nucleic acid will be degraded by the activity of these other set of molecules, like ribonucleases, right? And this is how it achieves an antiviral state, and the virus replication is blocked.
Right, sir. Got it. Thank you.
Okay, so summarize the innate immune responses. So we talked about innate immune effector mechanisms of your myeloid cells, your phagocytic cells, the activity of type 1 interferons, the activity of these defenses or antimicrobial peptides, right? And these mechanisms are also occurring in cell autonomous mechanisms, such as the activity of interferons, and the cells which have internalized these microbes, they can also undergo apoptosis, right? The recognition is occurring through these different TLRs, NLRs, and other innate immune receptors, right? As a result of it, so you try to control at the first level of defense these invasion by different microbes. This is not to say that if you adequately control these microbes by engaging your innate immune system, so you don't need to induce your adaptive immune responses, because the macrophages, APCs, and other cells which take up these antigens, right? So they can then engage specific TCR expressing cells, activate T cells. You generate different subsets of these activated T cells, T helper cells, T cytotoxic T cells. And in so doing, so you can engage the adaptive immune responses, production of antibodies, production of effector cytotoxic T cells or T helper cells. Right. So what time is it? 50. Okay, very quickly on this slide. Basically, you have these APCs, let's say dendritic cells, which have taken up these antigens, which can be the soluble product of these microbes, or apoptotic cells, or the other antigens are derived from these microbes. So those things are taken up by these APCs. These APCs then migrate from these peripheral tissue sites to the draining lymph node, okay, through these afferent lymphatics. So they get into the lymph node where you can find the antigen-specific T cells, okay, and this scanning has to actually take place, okay. So because in lymph nodes, so not every single cell that you will find will be specific to the antigen which are coming from this microbe and are displayed on surface of these APCs. Okay, maybe out of 1 million cells, so you have only 2-3 cells which are specific to that particular antigen. as they have rearranged their particular TCR during the development process, right? So this process takes some time to basically find out the best fit, right? Once that best fit is found, your APCs engage with naive cells of a particular specificity. Naive cells become activated cells, and once these activated cells are generated, so they will start dividing, and for a few days, so they will actually they will get retained in this lymph node, so that they can actually now make up sufficient numbers of theirs, and then they will start leaving lymph node, get to the infection site, and try to control it. So this is how basically you engage your innate immune cells with adaptive immune cells, right? Now, you can also induce B cells, right? For initial induction of B cells, you don't require the activity of APCs, because the antigen unprocessed can be sensed by the specific receptor present on surface of these B cells. Now, how do these antigens enter in lymph node? So again, through the same route. Okay, so these big antigens, or even directly the microbes, can actually enter into the lymph node, and that can activate your B cells. However, you require activity of your T cells to efficiently induce your antibody responses. Okay, so once you do that, for example, these are resting T cells and B cells, and once these cells are appropriately engaged by APCs, particularly when we talk about T cells, they will become effector T cells. They start making all these molecules, right? And carry out their function of killing. And then you also have these effector B cells that we call as plasma cells. And you can clearly see, starting from here to here, so the major thing that you see in these cells is a lot of endoplasmic reticulum. And the simple reason is because these antibody forming cells, or the B cells, have to make a lot of antibodies. Highly translationally active cells, right? So that is why, so you find a lot of endoplasmic reticulum in these B cells required for antibody production. Okay, so yes, so in next class, so we'll start talking about these things. What are immunogen, antigens, what is immunogenicity, antigenicity, what are the characteristics of an antigen, and so on and so forth. So now we are going to stop here.